All right. Thank you, Andrew Scholey, for coming on the Nootralize podcast.
Hello.
You are a researcher at Swinburne University, and you have done a lot of research on various substances and their effects on cognitive performance.
Correct.
Would you please start about alcohol, low-dose alcohol consumption? What are the effects of using small doses of alcohol on cognitive performance?
Yes, of course. I guess this work is pretty old actually, but it's an interesting phenomenon that within the context of the fact that alcohol is cognitively impairing, both acutely when people are intoxicated and during the alcohol hangover, of course, and also obviously longer term use of alcohol causes serious impairments to cognition and real brain damage. There is an interesting little counterintuitive phenomenon, which is that very low doses of alcohol appear to actually or can have memory enhancing effects. And I think I always have to, when I talk about this work, I always have to start with the warning and say that do not drink alcohol as a cognitive enhancer. That's really not a good strategy, but it's quite an interesting phenomenon. The way it works is that if alcohol is consumed immediately after being presented with some material that people learn, then subsequent recall is actually enhanced compared to a placebo. So we're talking about very, very low doses, much lower doses than you would that people tend to consume socially. And as I say, it has to occur in the immediate aftermath of being presented with the material. And there are two possibilities as to why this may occur. One is that that this low dose of alcohol you're presented with material and we're always updating our memory, and it may be that what happens is that the alcohol sort of interferes with that updating process, and so the material is kind of operated on for a bit longer, and so it's more likely to be remembered, if that makes sense. Or the other possibility is that alcohol is acting as a stimulant, and so it has a kind of reinforcing effect at those very low doses. And this is one idea about when people drink social, in any circumstances, when they drink alcohol has a strange kind of biphasic effect, as it's called. That is, there's a sort of in the immediate sort of when people first start drinking, this is talking about a single evening out, say, there's a stimulant effect very early on where people sort of report being a little bit lightheaded, and the inhibitions go a little bit, and they sort of become a bit more sociable, as people do with stimulants. And then that kind of peaks, and then there's a more of a depressant effect that kicks in. So it may be that this is tapping into that early stimulant effect of alcohol.
How large dose are we talking?
We're talking about, I think the dose is, so to 0.05 is the sort of legal drink driving limit.
Right.
In most jurisdictions around the world. It's a little bit higher in the UK and some other countries, and this is this is probably about 20% of that, 0.01, 0.02, so pretty low.
Yeah. Interesting that alcohol actually does have studies showing that if you do use it correctly, then it can actually enhance your cognitive performance.
Yeah, as I say, I don't think it's a good strategy, just because the dose and the timing are imperative. You know,
Yeah.
If you don't get those right, then the phenomenon goes. So, you know, it has to be after you've been presented with the material, and it has to be exactly the right dose. And actually, some unpublished data that we have seems to show that it may be related to that stimulant effect, because it's also is correlated to some degree with an increase in heart rate. There's a very slight increase in heart rate that occurs when people drink a small amount of alcohol.
Have you tried giving, or do you know of any researchers giving alcohol in about the same dose as we're talking about here, so a very low dose, when like slightly before exposure to the learning material?
Yeah, and that the effect just isn't there. So the timing has to be exactly right as well.
I see.
So you know, one idea is that when we form memories, there's this consolidation phase that occurs, and that this is sort of somehow helping that consolidation phase.
Right. So it's very, as you say, it's very dependent on when you use it and how much you use.
Exactly. Yeah.
Right. So it's probably not as you said as well, a good strategy for cognitive enhancement. Yeah. All right. Let's move on to glucose. Can you tell me a bit about your research on glucose?
Yes, I mean, that's something I guess that I've researched quite a lot, much more than the low dose alcohol, and over a much longer period. I think probably starting in it's probably about 20 years now, on and off. It's been a little bit of a hobby of mine. Yeah, it's a really interesting phenomenon, the idea that you can give people a glucose dose and it improves some aspects of cognitive function. And historically, this came from several different areas that kind of converged. So one was the idea that we know that the emotional material is remembered better than neutral material. So the traumatic events sort of stamp themselves onto memory more than neutral events. The example that's often given is that you know, you might park your car in the same car park every single day, and you know, if you say, well, where did you park your car? You know, a month ago, last Wednesday, you would have no idea.
Right.
But if on that day something very emotional had happened, so you know, maybe if you were assaulted, or as you're approaching your car, you saw that, you know, somebody had smashed the light, or even something else emotional that happened in your life, that you would remember the circumstances much better, and remember, for example, the location that you'd parked your car. So those are so that emotional memory involves a response that includes the release of adrenaline in the body, and adrenaline binds to receptors in the liver that release glucose. So researchers who were researching emotional memory wanted to see if you could sort of hijack that system. So instead of giving emotional memory, they just simply injected, in the first instance, animals with glucose, and found that they could enhance memory, and particularly if they injected glucose directly into the brain. And that led to some studies within humans. So the idea was that you can give people a glucose drink and you can enhance memory, and other aspects of cognition. And it seems to be, you know, and then at the same time, there was this development of research looking at the effects of nutrition on mood and cognition. So glucose was just one of the substances that was researched in that context. And that's really when I started to look at glucose. And the reason I think that this is probably a phenomenon that's pretty robust is that the brain is a very greedy organ. So it's about 2% of the average human's body weight, but it's always metabolizing at a high rate. So it's using about 20% of the fuels of the body, glucose and oxygen. And so, although there were a number of theories around at the time, myself and other people that I work with on this had this idea that maybe all that's happening is that you're just providing more fuel for the brain. So if you give extra glucose, then you can improve cognition because you're just really, throwing a bit more fuel onto the fire, if you like. And so we tested that in a number of experiments. Some of the experiments were with a PhD of mine, Mark Moss, where we thought that if we got this effect with glucose, well, how about if you give people oxygen? You would expect to get a similar sort of effect. And Mark had
because then they have more fuel.
Yes, just more fuel for cognition.
Right.
And so it was really just this idea that that if you so the brain, it's really interesting in that it's the most metabolically active tissue in the body, and yet it's got a sort of design fault in that it doesn't store any glucose. So a tissue like muscle tissue and the liver, which are also metabolically active, store glycogen, and the brain stores. I mean, it's got a tiny amount, but but that store is just depleted. For example, during sleep, it's got enough to last overnight. And so the sort of model that we developed was pretty simple. Actually, it was just a kind of supply and demand model. The idea that when you work the brain a bit harder, you recruit a bit more tissue within the brain. You activate a slightly larger area, or you activate, you increase the activation, the intensity of activation. And when you provide more fuel in the form of more glucose or more oxygen, you can improve cognition. And that has a number of potential sort of implications. One is that when you feel like you are using a lot of mental effort, that kind of real strain where you're multitasking or you're really putting a lot of effort into a mental task, that subjective feeling of effort is actually reflective of a real physiological process, real brain activation, which I think is quite interesting in terms of just the mind-body kind of problem. And that when you provide more fuel during those kind of experiences, that you can improve cognition more. And so this was, we tested it in a number of experiments, and it turned out that is the case. That as you sort of basically the tasks which we experience as being more effortful are the very tasks which are most susceptible to the effects of glucose. So that's sort of led to a nice model system really for looking at cognitive enhancement. Again, using glucose on a day-to-day level as a cognitive enhancer isn't a good thing, because glucose itself is, it causes all sorts of metabolic problems in the systemically in the body. But it's a nice way of just looking at the way the brain kind of can be improved. And in fact, some of our more recent work that I've done here in Melbourne in the last year or two has been looking at applying brain imaging and looking at patterns of activation when people are given glucose and seeing how they correlate with task performance.
So glucose research and glucose is very important for understanding how we can enhance cognitive performance through administration of different substances.
I think it's a nice model system. I mean, there are many ways in which cognitive enhancers work, but I think one of them is by providing fuel to the brain one way or another, either by increasing brain metabolism, so increasing the way in which glucose is sort of taken up into cells and converted into energy.
Right.
And that's certainly the case with another substance that I've done a bit of work on, ginseng. We know that ginseng increases the uptake of glucose into cells, and so that might be part of the way it's having its effect. But there are also other substances which improve blood flow to the brain, and again by increasing blood flow, either by increasing the diameter of blood vessels or the way in which blood is delivered to the brain. Any compound which does that has the potential to be a cognitive enhancer.
I see. How do you recommend that people use glucose? You mentioned people shouldn't use it like every day.
I mean, I personally, I think that glucose is more of a model system. It's a way of, just because there's a big literature showing this effect. So it's good for laboratory experiments. I'm not sure how effective it is as an everyday cognitive enhancer, just because of obviously the sort of amounts of glucose that you're administering are potentially, it's called obesogenic. So they cause metabolic problems. If you use these multiple times every day, it'd be like taking a bunch of energy drink every day, which is something else that I've researched, but more looking at combinations of glucose and caffeine. Just to see how they interact.
So not even like during a test, if someone is studying hard for a test and then they want to use it to perform better?
So there is a little bit of work from somebody else who's done a lot of work in this area, David Benton from Swansea University in the UK, showing that actually if you provide people with more of a kind of slow release of glucose, so if people are familiar with the idea of the glycemic index is a way of categorizing foods in terms of how slowly they release glucose into the bloodstream. It does seem that foods which have a lower GI or glycemic index maybe are a better bet for longer term sustained cognitive enhancement. But all of these things are acutely, they're just in the sort of minutes and hours following a single dose.
Right.
So and again, it's a bit like glucose and caffeine, which you found together in energy drinks. They can improve cognitive function, but there may be some downsides in some ways. And caffeine is another really interesting substance, because I was talking about how many cognitive enhancers appear to be effective by increasing the fuel delivery to the brain. Caffeine is actually is an example of a cognitive enhancer which doesn't do that. In fact, so caffeine works through a very well characterized pharmacological or chemical mechanisms. So we have these receptors on most cells of the body, including the brain, called adenosine receptors. And there's a natural substance called adenosine which builds up during the day, and when it binds to these receptors, tends to cause fatigue. And caffeine blocks these adenosine receptors, and the net result of that is a reduction in fatigue, because adenosine itself kind of blocks the release of physiologically arousing chemicals like the catecholamines of the brain. And so when you've got caffeine blocking it, it's a bit like, adenosine is like a key in a lock,
right?
And caffeine is like putting super glue in the lock, so you can't get the key in there, and so you don't get the effect of adenosine.
Right.
But another effect that caffeine has, which is really interesting, is that it's a vasoconstrictor. So it actually narrows blood vessels, and so that actually decreases delivery of oxygen and glucose to the brain, or decreases blood flow everywhere, including to the brain. So what you've got is two opposing mechanisms. You've got reduced blood flow. You've got increased kind of chemical markers of arousal, which is how caffeine has its cognitive enhancing and kind of alerting effects. And it seems that the kind of the pharmacology or the chemistry wins out in these two competing pathways, if you like, or processes. Now, what's really interesting about how we normally ingest caffeine is that it's ingested with vasodilators. So if you look at caffeine in tea contains things called catechins, which we know increase blood flow. If you look at caffeine in coffee contains these things called chlorogenic acids, which again increase blood flow. So another substance that's often used as a cognitive enhancer is guarana in South America, in Brazil in particular, and it contains catechins which increase blood flow. So does tea. Tea contains components which increase blood flow. So humans tend to ingest caffeine in a form which not only have you got caffeine in there, but you've also got these substances which oppose the reduction in blood flow, if that makes sense.
Right.
And so, I don't know if we've just evolved or somehow, have learned that those substances are likely to be effective cognitive enhancers.
Right.
But it is very interesting that they coexist. Another example is in cocoa, very, very source of chocolate. So
yeah.
Cocoa flavonols are vasodilators. They improve blood flow. So yeah, it's quite interesting that nearly every substance that humans use that contains caffeine contain these substances that improve blood flow.
Do you know if when someone consumes, for example, tea, let's say green tea, do they have more or less blood flow than before overall?
More.
More. So okay.
There's a lot of work looking at a phenomenon called flow-mediated dilatation. So this is when it's usually measured in the arm. So what you do is you put a strap on the arm. So you basically stop blood flow, and you can measure blood flow using a probe on the outside of the arm. And what you do is measure blood flow before, and you put this cuff on the arm, which occludes blood vessels, so it's the kind of restricts blood flow, and then you release the cuff, and you look at the kind of the rebound effect. So you can get a percentage decrease or a percentage increase compared to baseline,
Right.
depending on what your what the person is sort of ingested, for example. And there's a great deal of work actually by groups, particularly here in Australia, Peter Howe, Kevin Croft, Jonathan Hodgson, showing that when you do this, that tea improves blood flow. It increases this measure of blood flow both acutely, so a couple of hours after you drink tea, and chronically, so if people drink tea sort of over a longer period every day, you get these effects. And there's a lot of work showing that well, not a lot of work, but there's some work showing that tea improves alertness and cognitive function as well, and it's probably down to a number of components, but particularly a component called EGCG, which is one of the main sort of goodies within tea.
Yeah. It's very interesting. You mentioned that there is an individual variance in response to how people respond to different substances.
Mm-hmm.
How useful are the findings in studies where you researchers study a lot of people and then find average effects over the entire sample? How useful are they for individual people?
That's a really excellent question, and the answer is we're really just scratching the surface of this research. And one really important question is what makes people either responders or non-responders.
Right.
So there are so many individual factors that are involved with that. So genetic, talking about caffeine, for example, we know that I was talking about the adenosine receptor. We know that there is a mutation on the gene for the adenosine receptor called the ADORA allele, and we know that some people have a mutation which means that they don't get the benefit of caffeine. In fact, caffeine makes them anxious and jittery. And these people probably avoid caffeine, and quite rightly. So there are genetic factors, there are individual differences to do with things like that sort of history of nutrition, and obviously there's a lot of work at the moment looking at individual differences in the microbiome and how that might affect how nutrients are absorbed, or how they have their effects, because we don't know whether some of these effects are to do with how direct or indirect they are. So in other words, do we know if the substances we're looking at are crossing into the bloodstream and then crossing the blood-brain barrier, or whether they're maybe activating some component of the gut microflora, the bacteria in the gut, or just improving overall blood flow, which has a knock-on effect to the brain. So, it's a great question. We are talking about, of course, average differences, and that's why it's important to do studies which are powered enough to capture these effects. If they were, and these effects are, and I have to say, these effects are often quite fragile. I think what seems to be emerging is that improvements in cognition and mood are much more reliably found if you look at substances which have multiple effects. So This is the kind of absolute opposite. It's the antithesis of the way pharmaceutical drugs are developed. So rather than look for a drug which targets a single molecule or receptor or a pathway, there are certain substances which, particularly natural substances, which seem to have evolved with a whole cocktail of different effects, which seem to sort of nudge lots of different processes in a way which the overall effect is positive. So the answer is, these are very useful studies, and they provide, I think, a good evidence base, which is really important. But what we can't say is that, if we've done an experiment in a substance like, say, curcumin, and we found that people who are aged 65 to 75 show an improvement in working memory following curcumin, that doesn't mean we can take a 70-year-old and give them this dose of curcumin and guarantee that they will improve their working memory.
Right. It only means that they are likely to have those effects.
It means that they may be a responder, they may not. And there's a lot of work at the moment trying to understand what makes some people respond to these effects and some not. There's, one element of this which is quite interesting is something that we've been exploring at the moment, looking at sort of some methodological issues in the sort of experiments that we do in this field. And it's quite interesting that when you recruit for clinical trials which involve non-clinical populations, so we're not talking about trials to to try and modify a disease. They just shift, in my case, mood and cognition, and maybe other kind of health parameters, other health measures in the right direction. The people who volunteer for those trials tend to be not representative of the general population, so they tend to have a better diet, they're better educated,
right?
Probably got a bit more time on their hands, for example. And those people tend to be a little bit more high functioning anyway, and so maybe there's just a little bit of less room for improvement, if you like.
Right.
It's and that's very interesting because I think doing science in this area, you're always learning new things, and there are eureka moments. Some of those eureka moments just come with something that when you look back you think, that's so obvious, why didn't we think of that before?
Right.
But it's certainly the case that you need, obviously there needs to be some room for improvement.
Right.
I mean, if you look at some of the trials, for example, we've done trials in B vitamins, and it's a very controversial area, and sometimes we find very beneficial effects of B vitamins, and sometimes not, and other groups have found positive effects and sometimes not. So we're trying to really, you can either sort of look at those results and think, well, the results are mixed, let's just not bother researching this anymore. Or you can say, well, the results are mixed, let's try and work out why this experiment seems to have shown positive effects and this one
right
didn't.
Yeah. That really does make sense, that you try to find the reasons why the substances are effective for some people and not for others. If somebody listening wants to figure out if a substance works for them, we always say that they should listen to their body, listen to their mind, if possible, test their cognitive performance objectively, and assess if it is working for them or not. Do you have anything to add to?
And also try and, the other thing, I mean, one thing that's very difficult with self-experimentation is, there's a big placebo effect, of course.
Yeah.
But at the same time, people are taking these substances in the way that they would be taking them as cognitive enhancers, if that makes sense. As long as they're doing it in a safe way.
Mm.
With substances which are regarded as safe.
Right. Yeah, I agree. That is indeed very important. Ginseng. Tell me a bit about ginseng.
Yeah, so ginseng is, it's been used for many millennia, actually, as a kind of pick-me-up and a anti-fatigue agent. And yeah, it's got quite a long history of research, certainly sort of this century. And I've been involved with some of this, along with David Kennedy, who's at Northumbria, who was my PhD student, and Jonathan Ray, who was also a PhD student of mine. Really a series of experiments which were looking at whether ginseng had an effect or not. I mean, this is really how I got into this area, looking at ginseng and ginkgo biloba. And the reason was that I'd started to publish some of this work on glucose and oxygen, and a colleague and good friend of mine, sadly passed away very recently, Keith Wesness, brokered a PhD with a company, actually a Swiss company, who approached me and said, we've got these substances like ginseng and ginkgo biloba. Our customers are telling us that they feel much more mentally alert when they take these things. Your lab seems to be looking at cognitive enhancement, looking at glucose and oxygen at the time, and we'd like to sponsor some research. So I said, well, we'll take your money. Really, as long as, and I made sure we had this contract which said that when what I predicted was that we were just going to find a series of negative results. And I said, as long as we can design experiments that when we find negative results, we'll be able to publish them. Is that okay? And they were confident enough in their product that they said, well, go on then. And so these were pretty labor-intensive experiments where people came into the lab, on 5 or 6 occasions, and they'd take a dose of, say, ginseng, and they were tested, before they took the dose on that day, and then something like it was 1 hour, 2.5, 4, and 6 hours afterwards. So they were tested, on that day, and then a week later they'd come back and get another dose and go through it again. And so they were being tested, each person was being tested maybe 25 or 30 times, and there were 30 odd people, so it involved hundreds of tests. And what we found was, for example, one of the early tests with ginseng was that we did find a very significant improvement in memory. So these were high functioning, these university students who would come in, take their ginseng at, say, 9 o'clock in the morning, and 6 hours later at 3 p.m., their memory was still significantly better. So, really took me back, actually. I was shocked. But then I started, I was a skeptic. And then I started to read some of the animal literature and some of the early smaller case studies and other studies, and realized that actually there were very good reasons that that ginseng contains these, they're called ginsenosides, which are unique to ginseng, and they are very physiologically active. And so this memory effect has been replicated, several times by different groups around the world. So it's now a pretty recognized phenomenon. And we went on, or even in those early days, actually, we did some fairly crude neuroimaging showing that this was using EEG, where you have electrodes stuck to the skull, and they pick up the sort of chatter of the neurons underneath the skull. And we found activity that was related to ginseng compared to placebo. I should say that all of these studies are, the methodology we use is called placebo-controlled, double-blind. So we don't know what we're giving people; they don't know what they're getting. And there's a placebo, so everything's compared to something inert. And so compared to placebo, we found that ginseng showed this very specific pattern of activation, which is associated with improved working memory. So it seems to be a really robust physiological effect, which appears to possibly be associated with an increased uptake of glucose, because in another experiment we found that when we gave people ginseng, their memory, in this case working memory, improved. So when we were really giving them these effortful tasks, their performance improved, but glucose went down. So blood glucose went down. And so, we interpreted that as showing that ginseng was causing glucose to be taken up into cells. So it wasn't measured it was dropping in the blood and being available as fuel. I have to say, though, unfortunately, we then tried to replicate that finding, and we didn't find the glucose effect. Reasons I don't know. And again, that's research. Sometimes things just don't replicate, and unfortunately, we ran out of funding, so we've never been able to find out why that was the case.
Right.
But yeah, and so that was quite interesting. It went on to show showed a different. We showed a slightly different effect with an American ginseng. So there's the early work was with a substance called Panax ginseng, which is Asian ginseng, and then there's another sort of variant of ginseng, American ginseng, or Panax quinquefolius, which has a different complement. So ginseng, as I said, contains these compounds called ginsenosides, and different species of ginseng have different sort of amounts of the different. There's maybe a dozen ginsenosides, and the levels vary from one species to the next, and so they're slightly different in American compared to Asian ginseng. And we did find slightly different variants of or different cognitive effects. So Asian ginseng seems to improve long-term memory, where you sort of have to retrieve information that's you've encoded, whereas American ginseng improves working memory. So that is where you have to sort of hold information in mind. It's sometimes called it's like a cognitive or mental desktop, where you've got your information, you're manipulating it and moving it around.
Right.
And again, that's been replicated a couple of times. It seems to be pretty robust. And so so within something, you can't say that all ginseng is ginseng, because slight variations in the components of ginseng seem to impart different effects on cognition. And then the other sort of element to this is that often, unfortunately, some manufacturers will they'll sort of stretch the truth a bit about what's in their products. So I remember, when we were starting to do this research, I was in the US, and I went into the supermarket, and there was probably 20 different types of ginseng on the shelves, and it's interesting that people have done some experiments where they've gone into supermarkets. I think this was actually in Sweden, where they went to sort of health food shops and found different commercially available ginsengs, and they actually analysed them for their ginsenoside content, and found that some were very low, some didn't have any at all. So I think my tip is always to, if you can, is to find the compound that the research has been done on, because there's two reasons. One is that you then know that it's actually the same compound that has found the positive effects, because slight variants might make a difference in the effects. And then the other thing is you're actually supporting the companies that they're confident enough in their product that they are paying for research.
Yeah.
I mean, some people think the opposite. They think that if you're paying for research, then it kind of takes it somehow.
Yeah. But that is, of course, not the case if the research is done correctly, if the researchers are honest.
Yeah. I mean, we put all of our research on a clinical trials registry beforehand, so people know what we've predicted.
Right.
And then you can't go back and say, you can't just sort of go through it and cherry pick your results and say this is what we predicted all along.
Yeah. Right. And you mentioned earlier that many of these natural nootropics do act via many different mechanisms and have many different active ingredients as well that that lead to their cognitive enhancement effects. So, for example, ginseng has many different ginsenosides. Would you be interested, considering that you believe, it was my understanding at least, that you believe that it's the plants as a whole or the many different ingredients combined that lead to the effects? Would you be interested in researching a specific ginsenoside, for example, to see what the effects of that specific one ingredient is?
Yes. I mean, I think it would be very interesting to look at individual ginsenosides compared to all the ginsenosides, if that makes sense. So if you had, say you had 10 ginsenosides numbered 1 to 10, I'm just making this up, and and they were all sort of 10% of the whole, then you could have the ginsenoside number 1 at 1/10 of the whole sort of ginsenoside complement, and then compare that to the whole complement and a placebo. There's another another way that I think that this could be done, which would be even more powerful, but technically it's very difficult, and that would be to take an extract of ginseng that contained all the ginsenosides, and then one that contained all of the ginsenosides apart from the one that you thought was important.
Right.
And that would really nail it. So that would be like a sort of knockout psychopharmacology, but unfortunately we don't have the food science isn't far enough advanced to do that effectively. I mean, I guess one exception would be in the study of coffee and caffeine, where people have looked at the effects of coffee versus decaffeinated coffee. Now, that's really versus placebo. So there's a nice paper last year or the year before by Crystal Haskell, who's another of my ex-PhD students, and she found, I can't remember the details, but she found that there were some effects of, most of the effects were strongest if you look at caffeine. If you look at coffee, which contains caffeine, but there were some differences on some measures between caffeinated and decaffeinated coffee. But there are also some effects where there were beneficial effects of decaffeinated coffee compared to the placebo. And so that shows that, there are other goodies in coffee which can improve cognitive function, which are acting as a nootropic.
Right. Such as chlorogenic acid, perhaps.
Yes, we think it's probably chlorogenic acid.
Right.
Theobromine, possibly.
Right. So if somebody wants to experiment with any of these compounds that we've mentioned today, in order to figure out whether their experiments are successful, whether the substance that they are using is working for them, they will want to use a methodology that is as scientific as possible, of course.
Mm-hmm.
And you mentioned the problem of placebo effects earlier.
Mm-hmm.
Do you have any ideas regarding how people can control for that in themselves?
I think it's pretty tough,
right,
to do that. I would say that it's I wish I could give you a better answer. The fact is, because I'm a laboratory scientist, so I'm not a clinician at all.
Right.
I'm interested in things like how these effects can be applied to conditions like dementia, of course, and that's the ultimate goal of my research. But I think it's up to individuals. My only sort of caveat about this sort of thing is that there can be interactions with other medications. So people have to be very careful about drug herb medications, if you like, or herb medications. I think there are also effects where, for example, you look at, say, individual effects of substance A and substance B, and you think, well, look, if we could put these two together, this is going to be rocket fuel for the brain. And some which I've predicted very strongly that would be the effect. And when we've done the experiment, it's completely flatlined. There's no difference between the combination and the placebo. So and I think that's to do with these very complicated interactions. The other side of it is, so that's, I would just sort of say that people need to be careful in terms of self-experimental, although, the other side of that is that many of these botanical cognitive enhancers have been used for thousands of years sometimes. So we've effectively got centuries of the field studies, if you like, testing to their safety. And of course, the commercially available substances are, what's known as GRAS, generally regarded as safe. So this is GRAS certified. I think it's an interesting phenomenon because, I would be pretty confident that there is an expectancy effect and the placebo effect. That in itself is philosophically quite interesting because we know that the placebo effect is very real,
Mm-hmm.
In terms of things like pain relief. We know that if you, for example, if you block some of the opioid pathways in the brain with substances like naloxone, that the placebo effect disappears. So the placebo effect probably does involve release of endorphins or endogenous opiates.
Right.
You know, that's an interesting phenomenon, and I think it's, you know, we have this idea, particularly I think in the West or within Western phenomenon, that if you were in a trial and your say your cognitive function improved, and then you were told and you felt much more alert and you felt much more sharp, and your memory was better, your mood was better, and then at the end of it you said, somebody told you that you were in the placebo group,
Right.
you sort of feel like you were shortchanged,
Right.
or that you've been tricked. Whereas
Yeah.
actually, another way of looking at that would be, well, because I believed that I would perform better, or my mood was better than it actually was. I mean, that's pretty amazing, I think.
Yeah.
So and it is sometimes something that these experiments get criticized about, because we do a lot to try and make sure that everything is controlled, that the placebo is convincing, that there is absolutely no way that our participants will know which condition they've been in. But that's very different to real world, where people are taking certain sort of supplements or nootropics to improve cognition, because
Right.
they do it in the belief they'll work. So there's probably an additive effect of expectancy and the real physiological effect.
Hmm. Do you think that it could be useful to, if you have tested yourself, your cognitive performance objectively, let's say via some online testing site, that, let's assume for the sake of argument that it is a valid and reliable instrument for measuring cognitive performance online. Do you think that it is useful to compare, as a self-experimenter, your results that you have from a nootropic versus baseline on that online test versus other people having tested themselves baseline versus placebo in studies?
Yeah, I mean, that's a difficult question to answer. It's a very good question. Of course, the issue is whether you need to know your baseline. As you said, you'd need to know whether the test was valid, whether it was widely accepted as being valid. There are all sorts of challenges that we have with repeated measures using the same test. So is it a different set of stimuli? Are the stimuli randomized so that if there's some sort of, I don't know, let's say there was a word list, and that list contained a word, and that word kind of just kept, something that was, for some bizarre reason, was something that came up in the news on a certain day, over a certain period, so you're more likely to recall that word. So there are all sorts of controls that need to be put in place to make sure that there isn't some other external phenomenon. And I just think people need to be aware that, I mean, I'm all for people sort of finding out what works for them. But I think people need to be aware of the challenges and problems with interpreting this kind of thing.
Right. Yeah.
And I know that, for example, your podcast is very geared towards an evidence base, and that's the issue. You know, you have to make sure that things are evidence based.
Can you elaborate what you mean the issue is in what context?
Well, I think, you know, there are sort of all sorts of, I think there's just around cognitive enhancement, I've seen sort of certain, you know, for example, web forums where people are talking about certain substances where, you know, there isn't much evidence, for example.
Right.
So that's, you know, that's just the issue. But you know, I think that if people know, you know, maybe they know what's good for them. I mean, I think the other side of this, which is maybe, you know, maybe I'm sort of arguing against myself a little bit, but one thing that's quite interesting is that many of the substances that we find are effective,
mm-hmm.
the effects that we find are they line up very well with the traditional use of the substance.
Right.
So, you know, when we look at you know, Melissa or lemon balm, that was used as a calming agent in the 1600s. You know, mm-hmm. way before there were any clinical trials.
Right.
And so it can only have been because people were eating it as a herb and realized that they were feeling a bit calmer.
Right.
And the same for something like, you know, salvia or sage, where again, you know, that was something which was talked about in terms of its memory enhancing effects.
Mm-hmm.
And indeed, you know, our work shows that it does improve memory, not calmness. Whereas, say, you know, the indications, if that's the right word, historically have been supported by the science in the 20th and 21st century.
Mm.
So yeah, maybe, you know, maybe there are certain people who are very calibrated, you know, they're very good at detecting these effects. And I think that's probably especially the case when it comes to nootropic effects, because if you sort of historically in the 12th, 13th, 14th century, if you had a bruise and you know, took a plant and ground it up and rubbed it on the bruise, the right. bruise will go eventually.
Yeah.
And so it may or may not be to do with that. But if you take a substance or eat a food or a berry or something that that makes you feel a bit sharper or calmer,
right.
you know that, and you know that's something that's an immediate kind of felt effect. I mean, if you're a coffee drinker, you know that effect of drinking coffee, of you know, taking caffeine, and you know if you've had too much. So yeah, perhaps people are well enough calibrated.
Yeah, I think there, as you have outlined, are arguments on both sides. There is the placebo effect, but then there are also certain very distinct effects from certain substances. Mm-hmm. I really enjoyed this conversation with you, Andrew.
Me too.